Hello, my name is John Botilla, Senior Principal Scientist at Mina Therapeutics, and this is the presentation of our poster, In Vivo Induction of HBG for the Treatment of Sickle Cell Disease with Small Activating RNA. Normal adult hemoglobin, or HbA, is formed of a tetramer of alpha and beta globin chains. Sickle cell disease is caused by a mutation in the beta globin gene, or HBB, and forms sickle hemoglobin, or HbS. HbS irreversibly polymerizes in low oxygen conditions and causes life-altering symptoms, including vaso-occlusive crises. Induction of fetal hemoglobin, or HbF, where beta globin is replaced with gamma globin, is clinically validated to treat and prevent most complications of sickle cell disease, including VOCs. A level of 20% HbF of total hemoglobin is considered a protective threshold. Induction of HbF by activation of the HBG gene corrects the SCD phenotype by disrupting the polymerization of HbS and by diluting the amount of HbS present in red blood cells. Our approach to this problem has been to leverage our technology to specifically activate the gamma globin gene with small activating RNAs. Small activating RNAs, or saRNAs, are double-stranded oligos similar in structure to siRNAs, except that they increase gene expression in the nucleus rather than decreasing it in the cytoplasm. Once inside a cell, the saRNA will load into AGO and translocate into the nucleus, find its target sequence, and then associate with a complex of proteins that recruit RNA polymerase and increase transcription. Our cell system for screening saRNAs that activate HBG is primary bone marrow-derived CD34 positive cells that are erythroid expanded. After 5 days of expansion, saRNAs are delivered by nucleofection, and mRNA was analyzed 2 days later. This graph shows the results of screening 161 unique saRNA sequences targeting HBG. Lead saRNA sequences then underwent medicinal chemistry optimization to increase activity and stability and to prevent immune stimulation. The rules for these positional modifications were based on regression modeling of in-house saRNA-specific data. This led to the identification of development candidate MT011391, which had increased activity at both screening doses. Sequence specificity of this saRNA sequence was demonstrated by introducing mismatches in the 5' seed region, which negated activity. Global transcriptomics analysis with the chemically modified lead showed HBG2 as the most significantly upregulated gene with minimal off-target effects. The effect on globin protein expression was measured in primary CD34 expanded erythroid cells that were nucleoaffected and then differentiated for a further 7 days. The lead saRNA upregulates gamma globin an average of 11-fold in 13 independent donors, while also downregulating beta globin an average of 50%. This is indicative of an HBB to HBG locus control region switch. Dose dependency of the lead was evaluated in the same culture system, but here the expression of the HbF tetramer is detected by flow cytometry. The clinical benchmark for measuring HbF is as a fraction of total hemoglobin. Here, HbF induction was measured by HPLC in erythroid cells from SCD patients, where the lead induces to an average of 62% HbF, an absolute increase of 45 percentage points. This induction is superior to hydroxyurea, the standard of care. Percent HbF is tightly correlated with patient outcomes, but so is pancellularity. We therefore also assess the ability of the candidate saRNA to induce a pancellular effect in cells from both healthy and sickle cell donors. As shown on the right, we see a clear pancellular induction of HbF. In the clinic, we have used a liposome called NOV340 to deliver an saRNA targeting the gene CEPPA to myeloid cells for the treatment of immunosuppression in cancer patients. The CEPPA program established clinical proof of platform for the saRNA and liposome. We have comprehensive pharmacodynamic biomarkers, efficacy endpoints, and strong safety parameters in over 130 patients. Previously, we have shown that NOV340 delivers to approximately 60% of erythroid progenitor cells in the bone marrow of NHPs. To demonstrate in vivo activity, we needed an in vivo model where the HBG locus was active, as it is in SCD patients, so that the saRNA could further boost expression. Healthy NHPs have a closed HBG locus. However, F cells can be induced in NHPs with a combination of phlebotomy and hydroxyurea. As shown here in our pilot study, the combination of phlebotomy and hydroxyurea increased HbF expression to around 10 to 20%. This isn't very high in comparison to a sickle cell patient, but was enough so we could test proof of principle for the activity of MTL-HBG in the target cells. When we added MTL-HBG to this model, we saw an increase in HbF expression and an extended duration of effect in 2 of 3 animals. Finally, as there wasn't an in vivo model in which we could accurately measure the response of our compound in the context of an SCD patient, we used a quantitative systems pharmacology model of SCD. This model captures the process from erythroid progenitor cell development and globin gene transcription to RBC production and lifespan. When it came to modeling the features of MTL-HBG, we used conservative inputs, 40% delivery efficiency compared with the 60% observed in NHP, and a 3x increase in HbF compared with the 5x that we observed. The simulated output predicts that once monthly dosing with MTL-HBG is predicted to maintain the majority of patients presenting with a baseline HbF level of 5 to 10% above the 20% threshold. To summarize, we have identified a development candidate that activates HBG expression while also downregulating HBB in primary CD34 expanded erythroid progenitor cells. In erythroid cells from SCD patients, we see 62% HbF induction as a fraction of total hemoglobin. We have shown that formulation of this oligo in NOV340 has pharmacodynamic activity in NHP, and in SCD patients, this is modeled to induce protective levels of percent HbF. We are currently advancing this development candidate into IND-enabling studies. Thank you for your time.